Okay, everybody. Why don't we go ahead and get started this morning? My name is Andrew McDonald from LifeSci Advisors. It's my pleasure to host this breakfast for Pluristem and bring really a world-renowned key opinion leader here to present to you today. Today we have Dr. Sibai. He's Director of Maternal-Fetal Medicine Fellowship Program and Professor in the Department of Obstetrics and Gynecology at the University of Texas Health Science Center. He's a preeclampsia expert, and he's going to talk to you about the condition and about the unmet need, which I'm personally looking very forward to because I know very little about it. And then we will follow up his presentation with a presentation from the company. So with that, I'll turn it over to Dr. Sibai.
Well, good morning, and thank you for coming here. I'm really excited to see so many people are interested in preeclampsia finally, I don't know what generated all of this interest, but it appears as if now suddenly preeclampsia is moving at the top for investment. For some of you who really don't know me, I'm known as Dr. Preeclampsia. This is what people refer to me. I don't know whether it's bad or good. I have spent almost all my career working on this, and as you are going to see, it's a very serious condition, and if some of you had a relative, you'll really realize what I mean. Now, why is this really becoming suddenly important? I guess in addition to publicity on TV and some of the news program, there is no doubt this condition is increasing. And the rate is increasing particularly in Western countries. And why, we'll discuss this in a minute. I'm going to show you 3 different studies I participated in for the National Institute of Health. The first study was, we were doing a study in healthy nulliparous women. These are women who are in their first pregnancy. And at that time, we were trying to see if low-dose aspirin actually prevent preeclampsia. We studied 3,000 women. The study was disappointing. We didn't find that actually aspirin prevented preeclampsia. But at that time, the rate of hypertension and preeclampsia was about 12%. Several years later, it became that calcium might be important. So this is another study funded by your tax money. Thank you for your contribution also. We studied 4,500 women. Again, it was disappointing. Calcium didn't prevent preeclampsia. However, you can see how the rate went to 22%. And most recently, again, we tried to see whether antioxidants actually, particularly vitamin C and E given to women, will prevent preeclampsia. Again, it was disappointing and it didn't work. And you can see how the rate went up to 34%. So somebody will be wondering what's really going on. I think the 2 major changes that have happened in the United States and in Western countries are women are delaying pregnancy to later age. So this is really number 1. And the number 2 is the women in the United States and Western countries have been growing in the wrong direction. So, and really both of these are risk factors. So if you really think these 2 combinations, in general, the peak fertility for women is about 20 years. This is what I tell my daughters, afterwards it goes downhill because your eggs are not getting any better. So really, as women delay pregnancy to later age, their fertility is going to be reduced. And as a result, they are more likely to receive some form of assisted reproductive technology. And there is no doubt women who have IVF or some sort of assorted are also at increased risk. Again, I don't have time to explain to you the importance of sperm exposure, and sexual cohabitation. So really, if you put all of these together, obesity, you have multiple cystic ovary syndrome, you have more infertility, Obesity is associated with insulin resistance. So really putting all together, this is why the condition is increasing. So really, what will be the magnitude in the United States? If we assume in the United States we are somewhere around 4 million deliveries per year, and for this talk we're going to concentrate on preeclampsia that happen early. If we decide the 24 to 28 week are the group who will benefit from the therapy, the likelihood of this in 4 million deliveries is probably about 3 per 1,000. So you're talking about 12,000. As we expand this to less than 32 weeks, the number will be about 20,000. If we go to less than 34 weeks, the number will be 32,000. Now again, during the discussion, what I hope, if this really works, it will have implications to almost everybody. It's not only for the treatment. The next step will be to use for prevention. As I told you, we have been disappointed. We have spent more than $200 million of your tax money trying to prevent preeclampsia, and we haven't been successful. Now, why is this important? And particularly the early group that we're going to talk about. First, this is the group who are more likely to die, more likely to have stroke, pulmonary edema, which means fluid in the lung, liver hemorrhage, renal failure, admission to intensive care unit, and more important, long-term deficit. What we really know now, that pregnancy is really a window for the future, which means many of the problems that women have later in life might be related or identified during pregnancy. And having preeclampsia is really one of those that's becoming extremely important. Because of that, in 2011, the American Heart Association had put in their guidelines now, every doctor in the United States have to ask the woman whether she had hypertension or preeclampsia in her pregnancy. And these women are considered as a very high risk for coronary artery disease and stroke later in life. And they have to be followed more closely, like any other woman who's at risk for cardiovascular disease. There are more implications for the baby. As you know, the baby is what's going to keep humanity going on, unless we have cloning one of these days. So they always say, the babies are going to be the future of the whole world. And they are going to see again now there is something we call the fetal origin of adult disease. So many of the things that happen during pregnancy have implications what's going to happen to all of these babies as they grow and become adults later in life. So in addition to affect acutely, particularly in the form fetal death, fetal growth restriction, preterm delivery, hypoxia and acidosis, there are many complications that preterm babies have, which we'll talk about, particularly the babies we're going to talk about. And this is respiratory distress syndrome, but more important, the complication of that, which is bronchopulmonary dysplasia, which means these babies are going to be dependent on oxygen and some type of a machine all their life. And even if they are discharged from the neonatal intensive care unit, they are going to require multiple rehospitalization because of respiratory complications. Then some of them will have intraventricular hemorrhage, which means bleeding in their brain, and more important, the periventricular hemorrhage, which is really associated with cerebral palsy, which really has implications all their life. Then we have sepsis and necrotizing enterocolitis, which means injury to their bowel. So these babies are going to require surgery as a baby, and they might lose a huge part of their intestines. And again, this will have implications for all their life. Then, of course, the cost being in the neonatal intensive care unit. And finally, what are the long-term implications of these? As I said, these babies are going to be at risk all their life. So we're talking about what I call acute economic impact, but more important than the acute is the long-term implication. So what are really the manifestations? For some of you who don't know what preeclampsia is. Really, preeclampsia is a syndrome That's characterized by the first thing a physician will identify when the patient goes for regular prenatal care is the elevated blood pressure. And some of them will have some form of protein in the urine. The main reason prenatal care was introduced in the whole world is really all centered about detecting preeclampsia. This is it. Of course, over the years it has changed. But prenatal care was introduced in the whole world for early detection of preeclampsia. This is why every woman gets a measurement of her blood pressure and she gives a urine sample to detect urine protein. In addition, the patient volunteers certain manifestations because a woman will have hypertension, but she will not know she has hypertension. So she will develop some manifestations like having headaches, visual changes, some bleeding, and so on. In addition, these women will have some changes in their blood. And because of this, they are at risk for so many of the complications I'm going to show you. One of the complications is bleeding where the placenta attaches. And again, the placenta is the life for the baby. It serves like the lungs for the baby, the skin for the baby, the kidney. Once the attachment of the placenta to the uterine wall happens, then the baby is going to be hypoxic and ultimately the baby will die. And this really process could lead to the babies dying in utero within 15 minutes. Because once the placenta detached, then there is no oxygen anymore to the baby. And when it is severe like that, it not only has implications to the baby, this will lead to changes in the mother. So the mother have a condition called disseminated intravascular coagulopathy, which means she can bleed to death if we don't replace their coagulation or we don't deliver the placenta. And this used to be the most common cause of renal failure and need of dialysis in women about 40 years ago. But now with the change in health care, we have almost eliminated this. But this is still a major problem in really developing countries. The other thing we watch some is these women will have problem with their lungs. And this is why they will have a condition called pulmonary edema or other respiratory distress syndrome. And as a result, they require admission to intensive care unit. I think the most scary for all, if these women develop liver hematomas. And this is a very scary thing, not for you, for only any doctor, because most doctors have not seen this. And a lot of time when this happened in the past, it used to be fatal. So now we are seeing them, and we have mechanism how to deal with this. And again, this is a huge hematoma in the liver, and these women ultimately, they're going to require multiple surgery, but more important, some of them will require somewhere between 500 to 1,000 units of blood and blood products. I always say you need to get all the blood in the whole state, not in your hospital or not in the city. This is one of these patients where the hematoma even more severe and ultimately died after 700 units of blood and blood product. I'm sorry to show you this early in the morning during breakfast, you know. It's ugly. Now, this patient really was fortunate because her hematoma remained intact, didn't rupture. But you can see here just there is a laceration in the liver. We took care of it. And again, for some of these women, we can manage them expectantly, but they require a lot of support in intensive care unit setup. I think the worst thing is this, bleeding in the brain. Again, what we have noticed now in the most recent reports that are coming in the United States and the Western countries, that the likelihood of stroke is increasing. It's particularly increasing in women. And most of the stroke is happening related to preeclampsia, eclampsia, and 2/3 of this is happening after delivery. So this is becoming really a very important problem relating to this condition. Now, if the patient does not die, these patients are going to require, again, continuous care for many, many years to come. I mentioned to you before what is the implication of a woman having preeclampsia. This is the concept of pregnancy being a window for future health. What we know now, if a woman, and this is why the American Heart Association got involved in this, if a woman develop hypertension preeclampsia in their pregnancy, they are at very high risk for having chronic hypertension, type 2 diabetes, metabolic syndrome, coronary artery disease, cerebral vascular disease, and end-stage renal disease. More important, some of these women, because of the changes in the brain, they're having problem with impaired memory and cognition, and some of them will have post-traumatic syndrome. Now we come to the baby. I showed you really the implications to the mother. So really what happened to the baby? These are the implications when this preeclampsia happens early in pregnancy, which will be the subject of the talk today. If the baby is not delivered, either it's going to die in utero, or you are going to have abruption or separation of the placenta, or you are going to see reduced fluid, fetal growth restriction, and all of this will force us to deliver the baby premature. So really we have no choice currently. So what is the implication of having some form of therapy to prolong pregnancy? The first implication is survival. In the United States, and again the Western countries, we have done a long way now with survival. Babies born at 24, at 25 years, they survive. The problem, as going to see, they have serious morbidity. And what I'm showing you here, the implication of adding few days or a week in utero early in pregnancy. I don't know if this works. Do we have a pointer here? Mm-hmm.
The star on the remote.
Oh, thank you. Let's go back. If one look at survival, it is not a big deal to take a baby from 30 weeks to 32 or 34 weeks, because really the impact on survival is minimal. However, if you take a baby from 24 or 23 to 24, you can see how this curve is on the path. This is why these are extremely important when it relates whether the baby will live or die. As we go longer, as I said, a baby born in the United States at 30 or 32 weeks probably will have 99% chance of survival. It's not an issue. The issue is this. Even though they survive, you can see the impact on morbidity. Again, the morbidity, as I told you, is respiratory distress syndrome, sepsis, discolor, bleeding in the brain, and necrotizing enterocolitis. Again, as you can see, taking a baby from 23 or 24 weeks to 26 weeks has great implication in morbidity in this versus taking a baby from 30 to 32 or 32 to 34. So these early groups are very important, even if we can prolong gestation 4 days or probably a week. It will have great implication to the neonatal outcome and the cost, both acute and long-term. So really, all what we are talking about is this baby. And currently, as I said, you know, yesterday I delivered baby who has 450 grams, which is about 1 pound. These babies survive now. The problem is what happened to them. I have now 2 babies. One have been in the neonatal intensive care unit 1 year and 1 month. This is what really we're worried about. Yeah, we have done great now in survival, but there's a huge cost for keeping the baby now in neonatal intensive care unit, but what happened to them later on, particularly the implications for the family. They have to put up with this baby all their life. So what is the standard of care at the present time? What are we doing? These are actually my protocol, and these are recommendations for the Society of Maternal-Fetal Medicine, the American College. It really says that if a woman have this disease at less than 34 weeks, because currently if they are 34 weeks or more, we deliver them. We don't gamble. Because as I told you, the benefit for the baby might be slight, but the risk for the mother is high. In the early in pregnancy, the benefit is the baby is high, and the risk for the mother is high. But the mother is always say, I'm ready to take the risk to me, even if I die, if this will help my baby. So for those, if they are less than 23 weeks, or they are more than 34 weeks, we deliver them. For this group, and this is the group we're going to talk about, where the therapy will be beneficial. Currently, what we do, we give them corticosteroids, and this medicine we give to help the baby's lung develop. In case they are born premature, we are reducing a little bit the respiratory complications. We give them magnesium sulfate, and this is a medicine we give to protect the mother from having seizures or eclampsia. Also, magnesium sulfate has some neuroprotective effect for the baby's brain. And we give antihypertensive medications to lower maternal blood pressure to prevent them from having stroke or pulmonary edema. And then, in many of these women, after this happened, we deliver them within 48 hours. Because again, we don't want to take the risk to the mother. For some group, we try to do expectant management, which means try to keep the baby in utero as far as we can. The problem of what we are doing now, I always say, is barbaric. But this is the best thing we have to offer. Because what we are waiting to see, some catastrophe for the mother, baby, or both of them. But this is the best we can offer at the present time. So let's see what happened in those early in pregnancy. As I told you before, for those who happen at less than 23 weeks, we have not had a single baby survive, even though the mother said, I want to take all the risk. And you can see, these are the serious morbidity to the mother. Between 23.0 and 23.67, as you are seeing, I use even days, because early in pregnancy, days matter. For this group of women, because this is what we call the fetal viability in the United States, we are more aggressive for the mother to take risks. And as you see, maternal complications are tremendous, because we are trying to gain every day we can for the baby. And the survival is about 20%. And when we counsel with women, I have to tell them that among these 20% who will survive, at least 90% of them will have residual damage. So you are really talking about only 10% of those who survive will survive without any problem. As we go to 24 to 24.67, as you can see, survival increased to 43%, and maternal complications still about 1/3. And then every week counts, because you can see survival increase, and we are more likely to reduce maternal morbidity. So this is really a summary from a large data set to show you the relationship between taking a baby from 24 to 25 and mortality. And this shows you the morbidity and the baby surviving without morbidity. And you can see going from 24 to 25 or 25 to 26 have great implications for the baby. And these are some of the complications for the group we are going to discuss today. And this is what happened later in life in the form of cerebral palsy, impaired hearing and vision, and being disabled. And these are some of the things that the family have to live with. These are what I call complications. And almost most of the babies who survive and leave neonatal intensive care unit. So, why do we believe this medicine will work? Again, I don't want you to borrow this. These are some of the abnormalities that happen in women who develop preeclampsia. And this is the main reason I showed you a lot of things we tried to use to prevent preeclampsia before and didn't work. And what we think, these mesenchymal stem cells will have work on some of these. They're going to explain more to you in the presentation. This is what the PLX technology is. It's derived from normal placentas. Then these mesenchymal cells, after they are being extracted, they are processed, and ultimately we're hoping in the study to give to pregnant women. This is the mechanism after they are injected, and they will be injected intramuscularly. They are going to produce some signals that will have affect some of these pathophysiologic abnormalities at what I call the placental-maternal interface. And these are some of the markers or some of the vasodilator, or want to call them some of the toxins or vasoconstrictive agents that have been implicated in preeclampsia, hoping the cells will in one way or the other affect them. What's the data available? There is some data done by a group, from Texas at a different institution where I am. And this data has been generated actually in an animal model, which is really the rat, where the whole idea is to give this medication, and as a result in certain changes that are the pathophysiologic mechanisms of preeclampsia, which we can discuss during questions. And given to these rats, again, this slide has a lot of things on all what I want to show you is that when you combine pregnancy with this toll receptor 7 agonist and the PLX, you can see the changes in blood pressure. This is without the medicine, and this is with the medicine. It has effects on blood pressure. It has effect on endothelial function. Again, the main reason you are alive here, you need to thank your mother for giving you a very good endothelial. The endothelial cells really, we cannot survive without. They are really the lifeblood of everything we deal with, because they produce certain things that keep your blood pressure normal, they keep the proteins and the fluid inside your vessels. So the whole idea of preeclampsia is a process that leads ultimately to endothelial cell dysfunction and produces a cascade of events. And there is no doubt the animal data suggests that this really has great implications on placental vascular injury. And this is what we are hoping to achieve, that this study, if it turns to be true, is going to help either repair this or ameliorate the damage that's already there. So why do we need a trial for this? As I started, this is a very important clinical question, particularly regarding what is the optimal management of these women. Nobody really knows what to do. Everybody uses my protocol. I developed this protocol because I thought I can prolong pregnancy and help some of these babies, there is no doubt. But as I said, this is not the best way to go. It could change really current clinical practice for all of these women, not only in the United States, but all over the world. It has a huge economic impact, both acutely and particularly long-term. Again, it has what I call long-term implications to maternal cardiovascular morbidity and the fetal origin of adult disease. Thank you very much.
Thank you very much. What a great presentation. We can appreciate, how important this work is. Can you help me understand mechanistically, in your opinion, based on the cytokines that these cells may or may not be secreting, and when they're delivered intramuscular, what markers exist in the blood of these women that the cells that are IM injected can read as a change in the environment, and how do you know that the cells don't go the other way and exacerbate the condition? And I know that we don't necessarily know the answers, but from your understanding of the animal data, and then the other part of the question is, how predictive is the animal data in man and in that particular model? Thank you.
This is really a very important question. We have been arguing forever, can we extrapolate from the animal data to the human model? Unfortunately, that we cannot have access to the woman. We have to start somewhere. I think some of the whole discussion has to deal, is the human placenta the same as the rodent placenta? And even with the rodent placenta, there are differences between the mice and the rat. And the main problem in the human placenta is this trophoblast invasion that's extremely important for a woman to have a normal pregnancy. Without it, patient either will have miscarriage, preeclampsia, or fetal growth restriction. It requires deep invasion of the maternal vessels. And the rodents, the invasion is more of a superficial. The mice is more superficial compared to the rodent. At the present time, also there is a lot of agreement that some of these pathophysiology abnormalities might be similar in the rodents as compared to the maternal. And we will never go anywhere without these studies, so they have to be the first thing. And this is why the next step is to go and do the study. And this is the study is going to be a placebo. There will be a mechanism of monitoring. Now, from the data that's available about mesenchymal cells, we know that they secrete many of these proteins or novel markers, if you want to tell them, that go and affect, send signals beyond that. Now, do they go to the placenta or harm? From what's available data from others, it's not. And I'm sure they have some data they're going to talk about for some of the human data, not particularly in the pregnancy, what happened to these cells and so on. Ultimately, I think they picked up by the lung. This is where they really end up being removed. Correct me if I'm wrong.
Not in IM administration, only in IV administration they go to the lung.
Okay, how about the IM? Where do they go?
They stay in the area and they do not transient in the body unless the physician heats the blood vessel.
Because currently we're doing studies with these cells in babies actually to treat bronchopulmonary where they're given intravenously. So your question about safety is really extremely important, and this is why this is going to be really like a phase 1 type 2 study there. We can monitor this as we go along.
And the histological profile of women who are showing preeclampsia, is there a significant set of markers that you see, and that you see that across all of your patients, so that you can kind of see before they're actually in hypertensive crisis, you can see from their blood chemistry, their histology, that is it IL-6,
Yeah.
is it a high expression of TNF that gives you that warning sign?
What we see again from the markers we looked at, we have abnormalities in almost all the cytokines. Pregnancy, by definition, is the state of increased inflammation. You cannot have normal implantation if you don't have inflammation. And that all really starts by the deposition of the sperm in the vagina. It starts an anti-inflammatory process that ultimately leads to the process of implantation. Then if this really process does not work well, it produces what I call a relative reduction in blood flow and oxygenation. This will lead to secretion of certain anti-angiogenic factors. Then you have the endothelial injury, and it starts really this cascade. What we know is that there are two components that require to have preeclampsia. You need the abnormal placentation, and you need what we call constitutional factors in the mother. And this is where obesity comes into the existence. It's interestingly, women who smoke, smoking is protective against preeclampsia. And we really believe it might have to do probably with the carbon monoxide and the cigarette product. That's not the nicotine. So there is no doubt women who smoke, they're less likely, but we're not encouraging them to smoke during pregnancy. Because there are other complications. But what I have found in our studies I showed you about, if a woman smokes and she develops preeclampsia, she has the worst outcome. So again, there are a lot of what I call constitutional factors that come into existence. I really think the unique thing we're still trying to struggle with, it's a very immunologic process. It's like having a transplant. I always tell women, you are really receiving a transplant. And then it's like a tumor. You show it growing in front of your eyes every month. And it's amazing why some women can tolerate this transplant. Because really the transplant is the placenta, which is foreign to them. These are paternal antigens. So a woman will reject a kidney or a liver. Yes, she has to put up with a placenta, which at term is double the two kidneys and bigger than a liver. And this is where these immune phenomena play an important point. And this is what explains many problems in obstetrics, not only preeclampsia. It has to do with the women who miscarry, women who develop IGR, and some forms of preterm birth. And this is why I believe that if this works, again, I don't know whether it's going to work or not. This is why we want to do this. It might have implications beyond preeclampsia, because a lot of these other complications, they share a lot of these mechanisms I'm talking about. Yes, please.
Before we continue, I think we need to, for the sake of time, probably give one more question.
That raises an interesting point. Maybe the number is too small. If some women have had transplants, like a kidney or renal transplant, are they less prone? And say they're on lifelong chronic immunosuppressive. Is their risk for preeclampsia reduced?
You see, the problem with these women is how you define preeclampsia. See, if your definition of preeclampsia is hypertension and proteinuria, many women who receive a transplant, for example, if they receive a kidney transplant, they have already high hypertension to begin with. Then the immunosuppressive drugs they give them, they produce changes in their biomarkers. So yes, they have an increased risk of preeclampsia, but they have the risk that goes with hypertension and proteinuria. We don't know whether this is preeclampsia or this is really part of the medications and their transplant phenomena.
And a follow-on question. When you started your presentation, and thank you for the tremendous amount of information you provided, you talked about trying to prevent preeclampsia.
Yeah.
Now, do you see PLX cells in that scenario as well, or once you end up with proteinuria? And
I really believe this is where the future is going to be. I think treating only the condition is not going to be enough. It's not to be the best way, but we have to start somewhere. So if we feel that it's going to treat, the next step we're going to see, can we give it to the women before they develop? Because I think once the process of preeclampsia have started, and all of these mechanisms, the injury, I don't know whether PLX is going to work as ameliorating the disease or actually repair the disease. If all what they do is amelioration, then probably it's going to be more effective if we use it as a preventive therapy. But amelioration will be good because it will give us time to extend pregnancy by a week or 10 days. But this will not be the best way. Maybe it's too late to give it after all of this. And the implication is the market will be probably 100 times more if really ultimately being the way to prevent these women from having the condition.
Thank you.
Thank you very much. And thank you for your presentation. I'm sorry, but we're short because we have to continue on with the Red Show.
Okay. Again, thank you for the opportunity. And probably after hearing the complication of placenta, the natural question to ask us, how come we Pluristem enter into that dangerous space? And frankly speaking, the answer is very simple. We strongly believe that the fact that we collect cells from healthy births may help to prevent the complication of placenta. We have no rationale behind that in the beginning. We had no rationale behind that in the beginning, except one factor. In the critical limb ischemia study, a part of our patient entered the study with high blood pressure. About 50% of the patient that entered the study had the systolic blood pressure of about 160. And what we discovered, that many of them develop a constant reduction in the blood pressure, which gave us a clue that probably the placenta secretes something that generates a reduction in the systolic blood pressure. And if you've seen the model, the animal model that we used, and Dr. Sibai presented to all of you, what we have seen that when we inject our cells into normal animals, no impact on the blood pressure. Only when the cells have been injected into the preeclamptic animals, then we have seen the reduction in blood pressure. So there is an interaction, there is a secretion that is dependent on the condition of the animal and hopefully of the mother, as we have seen in our patient. So it's one. And second, in the end of the day, when you enter into complications like preeclampsia, you know two things. One, it's a multifactorial process. We at Pluristem believe that the disorders we should target in this stage of the cell therapy is complication which are known as multifactorial. And again, if I'm going to other fields, the AIDS treatment and now the immune therapy that is so popular, in all of them, we are talking about multifactorial treatment. You have to use a variety of drugs in order to deal with the complication at patients. So we believe that that's the rationale. And as we started the project 2 years ago, and we are now in discussion with the FDA to start. We applied the documentation, and we hope and believe that the FDA will approve the study, and it will start in US, and then it will continue as a multinational study with the goal to approve the product as soon as possible and to receive orphan drug designation and breakthrough therapy designation in the coming future in order to expedite the time for marketing. So we are, a few words about Pluristem. We are a biotechnology company traded in Nasdaq and in Tel Aviv. We work on the placenta, even though our technology can be used for other cells culturing, but we strongly believe that the placenta is a unique source, undiscovered source, and we'll discuss it later on. I think that we are unique in another aspect, that we work with many regulatory agencies. We are the only company in the cell therapy space that work with the FDA, European, Germany, Australia, Korea, and Israel, and we hope soon to add Japan into that list. So we are focused on talking and discussing the complication and potential of cell therapy with many agencies, and we believe it's very important to give you the feeling that we are working globally. We have about 165 employees, and we have about 30 patents that cover our technology. For the people that are worried about the money that we have in order to run the business, so as of March, end of March, we have 64 million in cash and cash equivalent, and the net burn rate a year is 25 million, so we have a few years of operation. The process is described here, and I think what is important, I'm looking for the arrow. Here it is. What's important to mention is the proceeding. So we start with birth, we collect the cells from C-section, and the main reason for that we have to have the mother to sign a consent letter that she donates the placenta, and we have to have a blood sample from the mother in order to test her for any viral contamination. Otherwise, we cannot proceed with the processing. In any case, during the manufacturing, in the first 3 weeks, we culture the cells in a quarantine to ensure that there is no contamination that have been carried from the mother to the facility, and only after that we release it for production. 8 weeks after the birth, we have the product ready to go. The product is cryopreserved in vials and stored for about 18 months today. The stability of the product is 18 months. We strongly believe that we can extend it to 3 to 4 years, but we have to prove it. We are working today with cells that have been cultured 3-4 years ago successfully, but we have to do all the testing according to the FDA guidelines, so we can release extension of those of lifetime only when we do the testing according to FDA guidelines. We develop a dedicated thawing device because what we discovered in cell therapy, that when cells are processed and then delivered to the site of injection, a lot of harm and damage is generated during the thawing process. Conventional way products are thawed today is not healthy for the product, so we develop a complete system from A to Z to ensure that the quality is kept. Mechanism of action have been mentioned by Dr. Sibai, and I will elaborate it a little bit. As you probably know, the common dogma in cell therapy was that in order to do a therapy, you have to inject the cells IV, intravenous, because the dogma was, or is, whatever one wants to say, that the cells have to arrive to the target in order to generate the therapeutic effect. What we discovered that it's not necessarily true. We are not against IV administration. What we claim that in IM administration we achieve a few benefits. One of them, we can inject unlimited number of cells into the muscle. IV administration, the one of you that follow IV administration of cells are aware about the first pass-through effect. Cells are stuck in the lung, which limit the number of cells that one can inject IV before a complication may occur. In IM, you don't see it. And second, which is more importantly, the cells last in the patient body for about a month. We cannot give you an accurate number because we cannot label cells injected to patient. So the cells stay about a month. During that time, they communicate with the patient body and secrete a cocktail of protein, which is modified according to the signal that the cell receive. Everything is proven in human and in animals, and the one of you that are interested, we can give you more data. And then when we inject the cells, we expect to have the therapeutic effect after a while, and we will see the data that's presented. To speak a little bit more about the mechanism of action in relation to the preeclampsia treatment. So we know, as we have heard from Dr. Sibai, that it's multifactorial. Most of the proteins that are known today in the preeclampsia are secreted by our cells, the VEGF and others. So we have a combination of androgenic protein, which is very important to ensure the cardiovascular interaction between the mother and the baby. And we have a cocktail of anti-inflammatory proteins, which are important to reduce the inflammation associated with preeclampsia. So we really believe that it may work. And what you see here is the mechanism of action of our muscle studies I will elaborate later. Cells were injected into the muscle, and then we documented the improvement 6 months following a single administration of our cells. We have a few clinical programs in space. All the clinical programs that are mentioned here are done with one product, with a PLX-PAD. PLX-PAD, PLX is for placenta expanded cells, PAD for peripheral arterial disease. And the rationale behind that we have been looking for indications in which the secretion profile of our cells is considered as a benefit for treating those indications. So we started with the CLI and IC. CLI, critical limb ischemia, IC, intermittent claudication. The program is out-licensed in Korea only to Cha Biotech, which is the largest biological company in Korea. The second program is the muscle and the orthopedic, the muscle and tendon. We announced the muscle data back in January. Even though we had only 20 patients in the study, we demonstrated 500% improvement in the muscle force, in addition to 300% increase in the muscle volume and nice p-values in the primary and secondary endpoint. And it's important to mention, because usually in cell therapy and cell-associated study, people are saying that in order to have a good p-value, a good significance, you have to have a large number of patients. And my answer to that is very simple. You have to have a significant improvement, and then you can reduce the number of patients that is needed in order to demonstrate good p-values. So we demonstrated it. We have the United deal. United Therapeutic is our first client. United is using our cells to treat pulmonary hypertension. They started phase 1 study in Australia, and we expect them to release some data later this year or beginning of next year. And the last program, as was mentioned, is a preeclampsia program. The potential market of all those indications is exceeding $27 billion, which is nice. Cha Biotech, for a few words about the deal. Cha Biotech is a deal in which they received right for Korea only. We keep manufacturing, we keep the IP, and upon establishment of the regulatory pathway and the approval, we will form a joint venture that will share the benefit 50-50. So it represents a deal with a high two-digit royalty number. United deal is completely different. United deal was done when we had no clue whether our cells can be used for pulmonary hypertension treatment. So it was $7 million upfront, 48 in milestone payment, and one-digit royalties. It was important for us to cut the deal 2.5 years ago, to prove that the business model is strong enough to convince the pharma company to enter. And remember, the business model is that we keep manufacturing, we keep the IP, and what we actually give to our partners is marketing rights for our product. We have another interesting product which is supported by the American government, by the NIAID, a part of the NIH. And the product here is scheduled to help patients that suffer from acute radiation syndrome associated with a nuclear disaster or a dirty bomb. We developed a dedicated product for the program. The product is dedicated for hematological deficiencies, and we are working hand-to-hand with them when we believe that in the end of the day it will lead to a product that will be stockpiled here in the U.S. and will be ready to treat patients in case something bad will happen. For some reasons, okay. A few things about the PLX cells. PLX, we call them mesenchyma-like cells, and there is a reason for that they do not differentiate, and they are supporting our mechanism of action that the cell should not engraft, but stay in the patient body, receive the signal, and secrete a cocktail of protein that may generate the change. We know how to use our technology in order to tailor the cell biology. And if one want to have a statement about what we are doing, we are using the epigenetic understanding. You know that there have been many arguments in the industry whether epigenetic is real or not. We prove it in our labs. We prove it in our culturing condition. We change the cell secretion profile just by changing the environment, changing the culturing condition, changing the scaffold, or other stuff. And we will really shortly a new product, all of them based on the placenta. You can see here the process in more details. So we start with the placenta, the one that we talked about. After 3 weeks, we have what we call the intermediate cell stock. For one placenta, we can have about 400 vials. 400 vials can seed 400 bioreactors. And then during the second stage of culturing, we can change the condition, the culture condition, and to have different products, the PLX-PAD that we discussed, and the RED, which is dedicated for hematological deficiencies. Okay, I will jump on that. Just to give you a feeling about how important it is, it's not only that we control the culture, and by that we can change the destination of the cells or the fate of the cells. It's very efficient from the economic point of view. 1 5-liter bioreactor is equivalent to 70 multitrays like that, which is used today by the industry. The marketing bioreactor will be 75-liter bioreactor and will be equivalent to 1,500 multitrays like that. And the importance here is to understand that the cells that are grown here on this tray is not necessarily the same as grown here. And think about 1,500 units like that you have to confirm bed-to-bed consistency. In the bioreactor, we can ensure it. And the best proof of that is that the FDA received our comparability document without any comment, and they approved our new facility with this unique manufacturing technology. We are located in Haifa, a little popular in these days when the Hamas is very proud when they hit missiles around Israel and so on. As I mentioned before, the lifetime of the product is, as of today, the shelf life is 1.5 years. So we have a stock of product out of Israel for 6 months. So in any case, if something happens, we can ensure the supply of our clinical studies. For marketing, we are planning to have a North American facility according to our agreement with United Therapeutics. Upon entering phase 3, we will start the build-up of North American facility to ensure the marketing supply to North America and other destinations. And probably the last comment here is a question that I would like to raise and probably to open the discussion later on. Can the cell therapy become the next generation of biological product? Can the cell therapy generate a company like Amgen, Genentech, Celgene of the world? And my observation on that is very simple. In order to prove it, we have to demonstrate significant clinical improvement in the patient condition, because there's no reason to claim for a paradigm change unless you can improve dramatically the patient condition. And we think that we have 2 clinical studies that demonstrate significant efficacy. We hope that preeclampsia will be added to that list and additional indications that we will do in the future. But that's the basic in order to claim that somebody is doing a paradigm change. The second one is to convince all the regulators around the globe that it's real, that it's safe, and that one can produce a product in a consistent way to ensure batch-to-batch consistency and so on. So we have the approval of many agencies. The next one is to demonstrate that you have not one product, not one cell source, and then you culture it, but you can take one cell source or more and culture the cells in a variety of different conditions and have different products. So we have it. We are the only company in our space that has its own manufacturing, and it ensures 2 things. One, we control production. Second, we can ensure high margin that is needed later on when you start marketing. And probably additional note on that, all the companies that have been mentioned, the Genentech, the Amgen, the Celgene of the world, none of them subcontract manufacturing to a third party. And even today, probably people of you have been heard, the Genentech is now in a process to acquire the Lonza facility in Singapore in order to have more capabilities, but not to subcontract work to Lonza. So I think it's something that is understood very well in the matured biological product industry. You have to control production to ensure quality and so on. Out-license deals is the best evidence that somebody is willing to pay in order to get some marketing right to your product. So we have 2 deals, and we hope to have more on that. And, of course, IP that was mentioned before. We protect our technology with 4 layers of protection. The first one is the 3-dimensional culturing. We generate a unique proposition of matter, unique characteristics of the cell. So the second layer is unique cells extracted generated during the culturing when the source is the placenta. The third layer is a therapeutic effect that can be achieved on unique indication like preeclampsia and others. And the last one is equipment that have been developed dedicated to the manufacturing processes and are protected. You can see here the data, and I will elaborate on that. We are talking about a phase 2 study in Germany in which we injected the cells for total hip replacement. And the rationale behind that was to test in objective manner the muscle force, the muscle volume, and the diameter of the muscle 6 months following the injection. So what you see here, day 0 was the injection, and the follow-up was done 6, 12, and 26 weeks following the operation and the injection. The green light represents the placebo. The dark blue represents the 300 million cell or the high dose, and the light blue represents the low dose, the 150. One question that has been asked by many people, how come that the 150 is doing better than the 300? And the answer to that we see it in other studies too. We have seen it in the CLI study, and other people have seen it in other clinical studies. Probably the understanding behind that is that we talk about cell-patient communication. If you inject too much cells, the communication is between the cells and themselves and not the patient. So the secretion profile is not good as, or not adapted to the patient condition as with low population of cells in the muscle. And I think that we are proud on that. As I mentioned, we had only 20 patients in the study. Improvement was 500%, and the p-value is very nice. The second study that we announced, and again proved the direction of efficacy with our cells, is a critical limb ischemia study conducted here in US and in Germany. And in that case, we had no control because it was the first in-human study, and the FDA and the Paul Ehrlich Institute did not accept our notion to have a placebo in that study. So we had a very nice number in the AFS. AFS is the number of patients that survive after 1 year. We had 85% survival compared to 64%, which is a Sanofi-Avanti study, the control of Sanofi-Avanti studies that have been conducted before. And a few last words about the real advantage of Pluristem compared to our to the landscape. So first of all, we have our unique manufacturing technologies, which give us a lot of advantages. Second, we think about a solution from A to Z. So we do whatever is needed, even though there are missing parts in the puzzle. So we design them, we build them in order to enable the technology to win, because we believe that in the end of the day, the only way to prove that cell therapy is real, that cell therapy is the next generation, is good clinical data. We receive a lot of support from the NIH. As one of you that follows the NIH decision probably aware that 6 weeks ago, the NIH announced a 10-year program to investigate the placenta. They arrived to the conclusion that the placenta is uncovered organ, and they're going to devote a lot of money and efforts to promote the research of placenta. We believe we will benefit from them. We believe that it is a good sign to us. 10 years ago, when we start to talk about the placenta as a source of cell therapy, we have been criticized by many people that we are not. And the placenta after 9 months is a dead tissue, so why to look for good healthy cells? And I'm happy that the NIH made a decision to support the direction. IM administration gives a lot of advantages. I know that it's a paradigm change. I know that people are used to saying that IV injection is the one that should be done. We believe that IM administration have a lot of benefits, as explained before. We have a real off-the-shelf product, no rejection, and the best evidence for that is that our cells, placenta cells, can be injected twice to the same person from the same donor, 2 weeks apart, 3 months apart, without memory T cells that are developed against the second administration. It cannot be done in other sources of human cells like bone marrow and fat, so we are unique on that. And the event that people are willing to hear about Pluristem, so we did a few achievements in the first 6 months. What we expect to hear in the following ones, first of all, the FDA decision about the Preeclampsia study, which we really hope that they will approve it. Around the Preeclampsia study, we can expect additional announcements like the open drug designation, breakthrough designation, and so on, but everything is conditioned with the approval of the study that we are waiting for. The second one is the start of the CLI phase 2-3 in South Korea. People are asking why we are delaying or why we are moving slowly on that pathway. The main issue is to define the final protocol properly. The first obstacle was the approval of the new facility, which was done in May. So now we are free to go into the study. The final discussions with the KFDA is underway, and upon approval, we will start the clinical study in Korea. Probably it will be extended to other countries. It depends a lot on financing. As a policy, we want to do advanced study, phase 2 and 3, with partners. So we have CHA in Korea to finance the phase 2-3 study in Korea. We assume that there is a chance that other companies from other places, not out of probably out of US, will be supportive to support the phase 2-3 study in CLI, and of course they will join the study if it will be possible. We expect to start to receive an approval for the additional orthopedic clinical study, rotating cuff study, and we expect to hear from United end of the year, beginning of next year, probably more probably beginning of next year, some data from the Australian study. Okay, I will jump how clever we are. That the management slide, and I like to finish all my presentation with that slide, and think the beauty about that statement, that a birth can generate therapeutic for many people. Let's hope that the placenta will be the proof of, the preeclampsia will be the proof of that. Any question, please? Thank you. Yes, please. Here.
Thank you. So, preeclampsia, do you think that you need to try PLX cells in healthy pregnant women before you move
No.
in?
We have evidence. Until now, we treated 100 patients, injected IM, with no significant adverse event. Second, we did a full battery of safety study at Charles River, in which we injected our animals to pregnant animals, and we took samples from the mother and the fetus to see if there is any harm, any unwanted manifestation of a complication, and everything was clear. So we believe that we don't have to do it, but you know that the last word is for the FDA. So we're waiting to hear from the FDA, and then we will answer. I do not believe that the FDA will ask for healthy women, but let's see. I tell you, you know what? I can give you the rationale, trying to think why. If you go to healthy women which are pregnant and you give them the PLX cells or any other cell therapy treatment, you don't know what may happen, but you increase the risk of complication because who knows what will happen. If you give it to preeclamptic women, they are in danger. So ethically, it's safer to give it to preeclamptic women than healthy women. That's my opinion, but let's listen to the FDA.
Okay, and I'm still not clear how you actually select the patients, the pregnant women with the condition. Is that based on hypertension and proteinuria or other measurements that you use?
The main measure will be the blood pressure of the mother. According to Dr. Sibai guidelines, mother will be measured 5 hours apart. And if they have two manifestations of blood pressure about 160, they will be considered preeclamptic, and then we will inject them following the conventional treatment. I think that the beauty of that study is that the mothers are in intensive care. They measure the blood pressure of the mothers every few hours, so we will have some kind of surrogate marker to let us know what's going on. Can we see a progress? Can we see a reduction in the blood pressure and so on? That will be the initiation. On top of that, we'll measure the proteinuria to make sure that the protein level is going down following the treatment. 24 hours after the injection, we see a reduction of about third of the. If we compare the blood pressure upon initiation of the preeclamptic condition and normal. So within 3 days, we see a full reduction, so it's about 1/3 in a day.
So based on some of your earlier comments, it sounds like it's a bit of a hunch when you first went into preeclampsia. I was wondering if since then you've gathered any evidence or even have any good theories about whether there's some sort of mechanistic implication for using placental-derived mesenchymal cells. Is there anything special about them from an efficacy standpoint in this?
I can tell you one answer in a very sensitive way. To answer that question, what we have tried to do the same experiment with Dr. Brett Mitchell using other cells. Bone marrow derived, fatty derived, many cells that you can think about. And luckily, we have seen that the placenta is doing the best. So not bone marrow, they do some effect, the other cells, but not as good as placenta, supporting our thesis that all the MSCs are not the same. You have to understand what they secrete. You have to understand that different cells do different therapeutic effects, secrete different cocktail of protein. So that's the only evidence that we have that placenta is better than others. And on top of that, as I mentioned before, the fact that we have safety data of 100 patients that receive PLX-IM gave us the support that we can enter into the preeclamptic treatment. The physician that came to me, one of the Israeli physicians that came to me 4 years ago after we documented the CLI good outcome, wanted to start to treat preeclamptic women in Israel. And I told him, no. On that time, we had only 15 patients or something like that. And so after a few years, after we have more confidence about the safety profile of our cell, we are more aggressive in our thinking. Yeah, George. One probably.
Speaking of different cells, could you be a little more specific about the source of the cells from the placenta? Is it from the baby side, the maternal side, and whether there's any difference in cells derived from either side that you've seen in some of the studies you just mentioned?
Okay, so let's start with the placenta. In the placenta, you have a few main regions. You have the decidua, which contains maternal. You have the villi, the chronic villi, the omnion, the chorion, the umbilical cord blood, and so. And we know that cells from each region are different from the others. So even you take the same placenta and purify cells from the omnion, or from the chorion, or from the decidua, they are different. So the raw material is different by definition, depend how you collect the cells. The second question is how to generate a different population of cells from the placenta. So the conventional way is to do a mechanical cut in order to take the region of interest and to produce the cells. We believe in different approaches, and the only thing that I say that the manufacturing process in all our products is the same. The beginning, how we collect the placenta, how we start the manufacturing processes, but then by culturing condition, we give advantage to different populations of cells. So when we decide that the first product is maternal product, mainly maternal, as a definition of the PLX-PAD is at least 90% maternal, we don't get those cells by taking the decedua only. We take from the area, from the region, and there by culture condition, we ensure that we have maternal cells, majority of maternal cells. The same way we go to other way around. If we want to get a product which is mostly fetal cells, we play with culture condition, and we generate a situation that we have a product which is mainly fetal, and so on and so forth. On top of that, we can change the properties of cells by challenging them. For example, if we grow cells under hypoxia, we see different secretion. If we grow cells under inflammatory condition, in presence of TNF or interferon gamma, we see differences. So there is a lot of room for changing. And again, all of the changes are what we call epigenetic changes. We play with culturing, culture condition, not inserting any vectors, any retrovirus in order to generate a change. More question, please. Can you give the microphone?
Could you use PLX cell therapies for different cancer types?
You know, today, if you say to somebody that you want to use MSCs or MSCs-like for cancer treatment, they will treat you as nuts because of the ability of those cells to secrete angiogenic factors and the common understanding that angiogenic factor can promote the development of cancer. Nobody will allow it. That's the common dogma. But there are other characteristics for the MSCs. MSCs have some targeting capabilities, so you can use MSCs as a vehicle to treat cancer. But it's a long way to go in order to do it.
Are you planning to do that in the future, perhaps?
We believe that it's one of the directions we should put our efforts in. Yeah, they have one. Yes, Boris.
Does this work? I guess it doesn't. So you're pursuing several indications, and certainly there is a number of potential biomarkers that you can monitor. I'm just curious if you have any kind of plan to do that, if the FDA is something that they've highlighted, and if that's going to be somehow presented or published at some point in time.
We are working on an article now which will demonstrate the mechanism of action in acute radiation syndrome. We spent about 2 years to understand the mechanism of action, and we believe that we have a good understanding, including knockout of specific protein to see if there is an effect or non-effect. I cannot elaborate on that too much because of publication issues. We want to send it to one of the top peer-reviewed journals, and we have to keep confidentiality to that point. I can tell you that we have a clear understanding in the acute radiation model. We have a good understanding on the PAD and the angiogenesis factors associated with IC and CLI. We have initial understanding about the preeclampsia. We arrived to the conclusion that in the clinical study, under the assumption that the FDA, of course, will approve it, we have a full battery of markers. We have been consulting with Dr. Sibai and others that we will test, taking blood sample from the mother, and hopefully by that we'll know better.
Sami, can you talk a little bit about...
If you don't mind, because people are...
Can we go back to the preeclampsia trial? So now as you're waiting FDA approval, I assume that you've submitted a design. Can you share elements of that design? For example, will there be repeat dosing? And how did you calculate the human equivalent dose based on animal models? And how will you be adjusting that? And I assume this is an open label trial, so you're going to be looking at kind of the first few patients in order to make adjustments. So is it a flexible design that you'll be able to make adjustments early on in the trial?
We start with 8 patients, open label 8 patients, to have a clue about the efficacy. Following the first 8 patients, it will be a 36-patient study. 2/3 will receive PLX, 1/3 will receive placebo. And then we'll have a futility efficacy analysis after 36 patients to see what is the outcome. And based on that, we will design the phase 3. The way we design it, it's a seamless design of 1, 2, 3. The assessment between phase 1, phase 2, and phase 3 will be done by an independent DSMB.
Sorry. So the idea is that after phase 1/2 data in 36 patients, the hope is that you can, with a small number of patients, go to a pivotal design.
Yeah, taking in consideration that to that point will be recognized as breakthrough therapy, right, and the open drug designation, which we believe we will get. So combining everything together, we assume we can do it.
Great. And can we switch gears a little bit in terms of the catalyst slides? Can we talk a little bit about why has it taken United Therapeutics so long to give us safety data, which I believe I saw projected for 2015? And can you help me understand what the next events are that we might see out of CHA in Korea?
You know, the good news about United that I cannot comment on that. We have an agreement. We are not allowed to say anything. What is going on, what is the proceeding, and so on. We have a promise that, again, they don't have to, it's not written in the contract. It's just a verbal promise at the beginning of next year that we will have data. So we cannot comment on that. The only thing that I can say is that IV administration is safe. We did, just to remind you, the United study is IV administration opposed to IM that we are using. The rationale behind that United believe that the first pass-through effect and the fact that the cells will be stuck in the lung for the first 36 hours will help the therapeutic effect. And you do not argue with your customer, and United is a customer. So they do it, they sponsor it, and we cannot comment on that. Regarding the CLI and CHA, everything will start upon approval of the study by the Korean agencies. As I mentioned before, we strongly believe that we will find additional partners which will be willing to support the phase 2-3 study in CLI, not necessarily in US for some reasons, but I cannot argue with that. So we assume that we will do it in more than one country, and it will be in additional countries. It will be done with partners that are willing to support the study in order to manage better the balance sheet.
Thank you so much, and we hope everyone at Pluristem is going to be safe with what's happening in Israel.
Thank you.
I'm just curious, the FDA hasn't approved stem cell or placenta cell therapy. I'm wondering how their attitude has changed from a couple of years ago to now, as it seems like more and more companies are in use, specifically on placenta cells, are getting into a therapy that might work.
I think that my answer is transparency. The FDA wants to see the data. It doesn't want to hear stories. And the best evidence to that, as I mentioned before, we built a new facility for cell culturing in Israel, the pictures that have been seen before, and we submitted to the FDA a full package of comparability study in which we compared the manufacturing in the old facility and the new facility. Not only that, we took two donors, two placentas, and we processed them in the two different facilities, and then we compared the outcome of that manufacturing, and the comparison was done, markers, secretion profile, potency assays, animal data, gene array, and we submitted all the documentation to the FDA, and guess what? We received an answer that you are approved. The same with the German, the same with Korean, and so on. What I'm trying to say here, that if you give the FDA the scientific rationale and strong evidence, they support you. If you come to them, for example, a third party will come to them and will tell them that, you know, placenta is safe because Pluristem did already a few studies. Please approve our placenta cell study for A, B, or C. The answer of the FDA will be no. On the other end, if the third party will come with the data package, they will approve it like that. So I like to work with the FDA. I do not agree with the notion that I hear from time to time that the FDA are nasty, the FDA are not open. They are good. They are doing the duty to keep the patient health. You have to support it with good rationale, and they are going with you. the preeclampsia study. I don't know the answer of the FDA, and I'm waiting. But we submit all the documentation, all the 2 years development that we have done to the FDA, and we are waiting to the judgment. We hope that we did a good work, and they will approve it. But if they will not approve it, and will ask for additional studies, we will do it, because we can understand the rationale behind that. We put a rationale, and we know that the FDA, whenever they answer you, if you give them a lot of data, they answer you technically. If you don't give data, put a statement, they can be nasty and give you other words. So open, transparency, and we like to work with agencies. And as I mentioned before, we have 5 agencies that approved our clinical studies already. Please. Probably, yeah. Thank you. Yeah.
See, I've met with the FDA several times in completely different ventures for preeclampsia. Everybody at the FDA, they're just eager and desperate for something for preeclampsia. So I'm not worried, because every time I go with them, they tell me, Dr. Sibai, anything that can be working, we are going to help. I think the FDA have been very, very good in this regard, because you are dealing with a group of women, which you cannot make them worse. They are as worse as they can be, you know. And this is why serious adverse events, which the FDA is very, very concerned about, and this was everybody will have a serious adverse event without treating them. So this is one thing is good. Now, for the FDA, when you want to give drugs to pregnant women, and some of you have really had great questions, you are giving these cells, where are they going to go? So this is really the most important question for them, that they're going to know, are these cells going to engraft somewhere? And in many of the other studies that were approved by the FDA, the trials I was involved with, one of their concerns, a lot of these medications, they're going to produce antibodies. So the FDA is going to see, will these antibodies will be produced in these babies, and what will the implications later on for the babies? I don't think it's going to be an issue here. We're not giving them a protein or something that produces certain antibodies to these agents. So these will be the questions the FDA is going to be looking at. Whether these cells are safe or not, the issue is that these are cells that are going to be deposited in the muscle of the patient. And their question is going to be what happened to them later on. Are they going to engulf somewhere else? Are they going to cross the placenta and go somewhere else? From what I heard, that these cells stay localized in the muscle, and this is where the convincing could go to the FDA, whether they will buy this or not. Mm-hmm.
Any additional question before we end up? So again, thank you very much. And I really hope that next time we will meet, we'll have some data from the preeclampsia study.